Thanks a lot, Tim. Yeah, you got a lot of that out right. I'll correct you on the keyword exosomes, which I find a tongue tangler at times myself. If we could just click forward a couple of slides. Just a little background on the company. Exopharm is a $71 million market cap company. It's been listed on the ASX for getting close to 2 years now. And we have three key technologies that are opening the door to a very important new area of medicine, which is exosome-enabled medicines that can deliver new types of cargo to new places in the body. Today, what I'll do is talk a little bit about the underlying technologies, what exosomes are, and give you an idea about why we all think and our investors think that Exopharm is such an exciting place to be right now. If you can go next slide. Okay, so what are exosomes? These are nature's way of passing nucleic acids inside of the body. What each cell in the body or any cell in a living organism produces are these little packets of material that protect very fragile RNA and fragile DNA from being destroyed as they move from cell to cell. And the reason that organisms need exosomes is so that cells can collaborate and start to work together, so that a muscle cell next to another cell, that cell knows that it needs to be a muscle cell too. If you look at the material that make up an exosome, there are three parts. One, there's a package that holds everything together. Secondly, there are these proteins that sit on the outside of an exosome, and they are addressing materials. They help tell the exosome where it needs to go, and it also tells the cells whether or not to take up the exosome that's nearby. And then inside is this raft of things, this RNAs or proteins or fats. These can be building block materials, but they can also be instructions. And the instructions are the really important and exciting part of exosomes, and it's why this is becoming such an important field right now. Next slide. Thanks. What's funny about exosomes are that even 14 years ago, they were considered the rubbish bin of the cell. So basically, the cells were packaging things they didn't need up and throwing them out. And that was how the cells cleared their bodies. But in actual fact, once we realized that they were actually forming this technology that allowed cells to communicate with each other, it became obvious how powerful these could be if we could harvest them and access them for medicine delivery. And really over the course of the last 5 to 7 years, it's become super clear that if we want a natural way to deliver targeted medicines in the body, these biological medicines, then exosomes or their counterparts would need to be involved. Next slide. So Exopharm as a company looks at this problem as 3 steps, really. One is to establish a technology platform that we can use to make exosome medicines, because this is something that's being done for the first time. Secondly, once we have a platform, we want to prove it out using clinical evidence to demonstrate that we can purify and scale up those technologies to something that could be used inside of a medicine. And then ultimately, and our clear goal is to take that technology and use it to make new medicines using exosomes. The company itself was founded on day one with the concept of producing high-scale, economical, consistent ways for exosome or EV. I'll use EV as a substitute for exosome sometimes. An EV medicine. We want to be able to scale it up to treat thousands or millions of patients. We're not interested in treating 5s or 10s of rabbits, right? We need something that will do a lot of this work for us. Next slide, please. What we've assembled over the course of the last really 2 years is this important portfolio of technologies. The first one, which we grew in-house and invented ourselves back in 2016, is something called LEAP, which is a scalable process for purifying exosomes from a whole variety of biological materials. And then the second 2 are technologies that we were able to bring in license from US inventors last year and bring on board to do some really important work for us. The first we call EVPS, like GPS, which helps us identify where exosomes will be taken up in the body. We can change the proteins on the outside of exosomes, as I'll describe a little bit in the future, and get them where we need to go. And then the last technology, which we call load, is a technology for really enhancing the quantity of medicines inside of the exosomes we create. Let's step through these technologies. Next slide, please. Yeah. So what's most important for you to understand about exosomes is what's holding back their use is not really understanding of them at this point. It's really, how do we make something that's scalable, something that can treat thousands of people? And there's only one technology that's ever been described that can do high volume purification of exosomes, and that's the technology that we invented and own, that we call LEAP. LEAP is a technology that uses cation exchange chromatography, which is used as an industry standard process across pharmaceuticals. What we've done is uniquely adapted for the use of EV purification. It's a proprietary process, and it's something that's already been patented in Russia. We've got a patent that's in a fast track in the US. And we think that this is going to be an absolute game changer and an essential component in any company who's using exosomes for medicines. Next slide. Let's talk about something that we're all near and dear to our hearts right now, which are the vaccines. Pfizer's vaccine, which we have had here, and Moderna's, which we haven't, is based on the idea of taking RNA from the virus itself, wrapping it in a little blob of fat, and causing it to, putting it into the body, and teaching cells to create parts of the proteins that the virus would create themselves. And by doing this, we're training the immune system to find the proteins, to fight them and destroy them, which gives us the immunity that we're looking for. Now, the reason that they're wrapped in LNPs is because without LNPs, lipid nanoparticles, or this little blob of fat, as I call it, that RNA would degrade long before it got inside of a cell. The problem is that these LNPs are artificial. They're toxic. They cause an immune response. They don't necessarily get to the cells that you want them to get to. But really what they're doing is acting as a pseudo or an artificial exosome. We're protecting this RNA so that it can get into a cell. Obviously, this isn't the right technology for most indications. It works well enough if you have one or two treatments, like in a vaccine. It works fine if you inject it straight into a muscle. But if you're trying to treat something like glioblastoma, for instance, in the brain, or some other disease that involves getting into different cell types, you're going to have a lot of limitations. And if you go to the next slide, I really encourage you all to join us next week, next month, sorry, it's next week, where we're going to talk about these LNPs and delivering RNA medicines, and overcoming the challenge of delivery, not just using LNPs, but using exosomes, because we think that this is one of the key areas that exosomes will be first used around the world for medicine. And the reason that it's so important, and the reason it's useful to compare ourselves to lipid nanoparticles, or LNPs, is that the exosomes by nature solve a lot of the problems associated with LNPs. They're not toxic. The body doesn't reject them. They can be dosed frequently. And until the only thing that was holding them back really was scalability. And that's what that LEAP processing that I described before, that's the thing that we unlock. And so by providing this capability, we now suddenly have the ability, and any of our partners who license through us have the ability to produce high-scale exosomes, which then allow them to potentially treat a whole host of diseases that would not be available if they just relied on LNPs. I'm going to shift now to the second technology that I mentioned before. This is called EVPS. Yeah, thanks for the slide. And EVPS is a way that we change the proteins that are expressed on the outside of an exosome. And that's super important because it's those proteins that determine where the exosome is taken up by which cells. So if we can control the proteins on the outside of an exosome, we control where the exosome will be used, and we can control where the cargo gets delivered. If you go to the next slide, thanks. This is a really interesting example. And what we've done here is stolen something from the coronavirus, which is the protein that sits on its outside. It's called the spike protein. You might have heard of it. And we've actually put that into a human cell line so that the human cells will express that protein on the outside of its surface and on the outside of exosomes that are created. And we can see that in these images. I won't go into the details around that, but basically what we're doing here is demonstrating that a targeting protein, the one that's used by coronavirus, can be attached to an exosome of any type. And next slide, please. And one of the important reasons that we're doing that is that it's actually really hard to do. It's hard to do because the spike protein itself is massive. And there are a whole number of technologies that we can use to target exosomes to specific cell types that involve different types of chemistry and different types of biology, such as nanobodies, cell-penetrating peptides, and so on, that are much, much smaller and therefore much, much easier to express on the surface of a protein. So this is really interesting and useful evidence for us as we talk about partnering with other companies. Next slide, please. The third technology, which we call LOAD, is a way for us to localize the RNA. Like I mentioned, the RNA is really the cargo that we're interested in a lot of cases. And by taking this LOAD technology, we basically tag the RNA for being packaged into the exosomes. And by doing this, we can demonstrate that we get more and more capability, more and more power into a given exosome. And next slide. And when we put all three of those pieces together, what we can demonstrate for our partners and potential collaborators across the world, the pharma partners that we're talking to every single day, is that we have a technology set that allows us to not only produce the type of messaging that we want on the outside, the cargo that we want on the inside, but to do it at a massive scale. And this set of technologies can be applied to applications across the body: rare diseases, cancers, viruses, neurological diseases. And it's a very, very exciting time for exosome medicine. And it's because of technologies like these. Next slide, please. We've assembled a large team, a really capable and experienced team that has know-how from a whole range of global leaders, both in business and in biomedicine and technology in general. We're very excited about the technology and the people that we have. And when I go into discussions like I do virtually every day with potential partners around the world, we see that enthusiasm reflected back to us as well. So with that, I'll say thank you very much, and I'm happy to take a few questions, Tim.
Thanks, Chris. That's it really is quite fascinating. Can you tell us a little bit about the patent and where that is in terms of the time frame, and what does that patent actually protect?
Right. So I just want to be clear. We probably have about 6 patent families at this point, but the one I think you're referring to is LEAP. And LEAP is a very, very broad patent application, because using cation exchange is going to be the gold standard for purifying exosomes. It is used in industry every single day for a whole variety of techniques. It's highly scalable. The resins themselves are washed. There are fantastic suppliers around the world, but what wasn't known was how to take them and use them for exosome purification. That's the scope of the patent, and it covers patent resins that are made by all the large companies of the world that are in this space: Merck, Cytiva, Sartorius, Paul. We have a patent application that will cover all of that for its use for exosome medicine. So what we've seen so far is that the claim, every one of our claims went up in Russia. We've gotten some recent feedback from the States as it's going through a fast track. We feel very positive about the conversations we're having with the patent offices. And really, once those patents are in place, what it does is it facilitates us in talking to our peers, really, in the exosome medicine fields, as well as other pharma companies, and helping them unlock the power of these technologies for their own processes. We would love to see, and we think eventually it will become a standard for all exosome medicines around the world.
And there's quite a technical question here in regards to, so with the phase 2 trial being successful for Israel's exosome CD24, how are they planning on producing the treatment on mass scale without the use of your LEAP technology?
Well, that's a fascinating question, Tim. You know, effectively, and trust me, this is probably the the place where we've spent more time doing due diligence than any other for the last 4 years, is what other technologies can be used alongside cation exchange. And realistically speaking, the best technology for doing that is hiring lots and lots and lots and lots of people, lots of PhDs, lots of medical scientists, getting them to basically create a sweatshop for exosomes. We're not going to do that. Our technique is a way we did a clinical trial where we processed 50 doses that's all we needed for the trial in 8 hours with 2 people. If we wanted to produce 500 doses, it would still take 2 people 8 hours. If we want to do 5,000 doses, 2 people 8 hours. That's what we mean by scalability. There's no way to do that in a phase 2 trial or a phase 3 trial. It's fine to run a phase 2 trial. If you're willing to spend the money and you find enough of the sweatshop, you're welcome to do it, but it's not something you're going to do at phase 3, and it's not something you're going to do in production, of course.